Imagine a tiny organism, invisible to the naked eye, curing millions of people worldwide of devastating diseases. Sounds like science fiction, right? Meet ivermectin, a life-saving drug discovered in the most unlikely place, a scoop of dirt from a Japanese golf course. This extraordinary discovery not only changed global health but also earned its discoverers the prestigious Nobel Prize in Physiology or Medicine in 2015. But how did this humble scoop of dirt leads to a miracle medication? Welcome to Fascinating Drug Discoveries. I'm your host, Dr. Shugufta Wasim, and today we are exploring the incredible journey of ivermectin, a game-changing drug in the fight against parasitic diseases. Ivermectin's story began in the 1970s with Dr. Satoshi Omura, a Japanese microbiologist specializing in searching for microorganisms with medicinal properties. From a humble soil sample collected near a golf course, Omura isolated a bacterium called Streptomyces ivermectilis. Recognizing its potential, he sent it to Dr. William C. Campbell at Merck, who identified the bacterium's ability to produce compound with remarkable antiparasitic properties. What made this bacterium special was its ability to synthesize ivermectin, a compound with potent antiparasitic effects. Ivermectin is a macrocyclic lactone, featuring a complex 16-membered lactone ring with double bonds and a disaccharide sugar moiety attached, making it highly effective against parasitic organisms. Dr. William C. Campbell and his team modified ivermectin structure to improve its safety and efficacy for humans and animals. This led to the creation of a derivative we now know as ivermectin. Ivermectin is derived from ivermectin through the hydrogenation of 22-23 double bond. Initially introduced in the 1980s for veterinary use, ivermectin quickly proved to be a game changer in animal health, protecting livestock from devastating parasitic infections. But its true potential became clear in 1988 when it was first tested in humans. Ivermectin showed exceptional efficacy in combating onchocerciasis or river blindness, a parasitic disease that causes severe infection, skin lesions, and blindness, particularly in sub-Saharan Africa. Ivermectin's ability to kill the larval worms responsible for the disease and halt transmission was nothing short of revolutionary. But ivermectin's story didn't end with river blindness. It was soon employed to combat lymphatic filariasis, another parasitic disease causing debilitating swelling and disfigurement. Ivermectin has been a cornerstone in treating river blindness and lymphatic filariasis, drastically reducing the burden of these diseases worldwide. A single annual dose can disrupt transmission, offering a practical and effective solution for mass treatment programs, especially in remote areas. So how does ivermectin work? It targets the nervous system of parasitic worms, binding to specific ion channels known as glutamate-gated chloride channels in parasites, paralyzing the worms and ultimately killing them. Ivermectin disrupts the nervous system, leading to paralysis of the worms. What's truly remarkable is its selectivity. The glutamate-gated channels are not present in vertebrates, making it highly effective against parasites but safe for humans. It's this balance of safety and efficacy that makes ivermectin such a remarkable drug. Ivermectin's story is one of innovation and compassion. Merck's decision to donate the drug free of charge through the Mectizan donation program transformed it into one of the most successful public health initiatives in history. Since its introduction, the program has distributed over 4 billion doses of ivermectin, drastically reducing cases of river blindness and lymphatic filariasis. In 2015, Dr. Omura and Dr. Campbell were awarded the Nobel Prize in Physiology or Medicine, recognizing their pioneering work in developing ivermectin. Their discoveries serve as a reminder of the untapped potential in nature and the power of scientific collaboration. Recent studies suggest that it may inhibit tumor growth by interfering with key cellular processes such as mitochondrial metabolism and signaling pathways. While these findings are still in the early stages, they hint at an exciting future for repurposing ivermectin in oncology. Various researchers have shown that ivermectin regulate glucose and cholesterol level in diabetic mice. Like all medications, ivermectin is not without its side effects. Most are mild, including dizziness, nausea, and diarrhea. Rarely, individuals may experience serious reactions like low blood pressure or allergic responses, particularly when treating conditions with high parasitic loads. This is why careful dosing and medical supervisions are essential. The journey from ivermectin to the ivermectin is a story of persistence, creativity, and the transformative power of science. It's a shining example of how a humble bacterium in soil can lead to a global medical breakthrough, saving millions of lives. Ivermectin truly earns its place as one of the most fascinating drug discoveries of our time.